Okay, let's continue with our class. So this next figure we saw in the textbook, it's worth explaining a little bit because it's maybe difficult to understand. But basically, it's an experiment, again a positron emission tomography experiment, where subjects are presented with a screen, which is the one we see in image 1, and in this screen we can see several number of squares with they have this characteristics. They have shape, they have color, and they move. So the arrows indicate in which direction they are moving on the screen. So the experimenter is able to change any of these characteristics. So they can change the shape, they can change the color of the stimuli, or they can change the way in which they move. And they will be asking the subjects to pay attention to either one of these characteristics or to all of them. So subjects will see very briefly, also this is very important, they see very briefly the stimuli on the screen, and then they have a delay period, and then they see again stimuli but in different shape or different move or different color, and they have to just say if they are different in any aspect, so they can rely on one of these characteristics or all of them. And when they make the experimenters make the subtraction of the brain activity between one paying attention to one characteristic or all of the characteristics, they will see different activations in specific brain areas. And this is what they found. This is actually they made a new image where they just paint with color every brain region in where they found activation. So we can see in here different areas of the cortex, and those areas are showing, those spots are showing higher activity when different attributes of the stimuli were being discriminated. For example, whereas the ventromedial occipital cortex was affected by attention in color, this is what you see here with blue colors, and shape, which are the orange spots, discrimination task. This was not affected in the speed discrimination, which is the green spots. And on the other hand, whereas in parietal cortex we see the influence of attention into the motion, there is no influence by the other characteristic of the task. This is what this image is showing. The next one is we change in here so far we were explaining experiments with humans and what we call non-invasive techniques like positron emission tomography and magnetic resonance imaging. And here we are changing to experiments with monkeys, and they use for this experiment single unit recording, which means putting an electrode into the brain and register the action potential from specific neurons. So you put the electrode in one particular region in the brain and you see what happened with those neurons, one neuron or several neurons in that specific area. So this image, figure 21.10, is showing the experimental task, which consists of the monkey sitting in front of the screen. They have to fixate at the center of the screen, and then they will see one target stimulus flashed either to the left or to the right of the screen, and they have to move their eyes from the center of the screen towards the stimulus. This eye movement is called saccade. So this is a saccadic movement, when they move the eye from the center to where the stimulus was projected. And here we can see what happens when the monkeys, when the stimulus appear in what they call the receptive field. This is an important concept. Before doing these experiments, the experimenters put an electrode on one specific brain area, and then they show stimulus at different points on the screen, and they see that somehow the neurons respond to one specific area inside the screen. So, for example, 90 degrees, 45 degrees. When you put the stimulus there, the neurons respond selectively to that specific space. So we call that receptive field. And then once they found out where the receptive field for that neuron is, they use that to show the stimulus there or at different places. The monkey always is looking at the center of the screen, and then they show the stimulus, it disappears, and the monkey has to move the eye towards where the stimulus was presented before. And what they found here, and you can see in this figure, is in green color you can see when the stimulus is on. So this vertical line illustrates when they turn it on, and it seems like it remains on. And down there in pink color we can see the cell activity. So every vertical line is illustrating action potentials. So you can see action potentials and down there in the horizontal line you can see that the eye is not moving. And then down there, the stimulus is still on, there is an increase of action potentials, and the eye moves towards the stimulus. They make a saccade. This is what this figure is illustrating. And here, again, figure C, showing the same, but in this case, the eye, you can see here, the eye is moving towards the opposite to the receptive field, and you can see that anyway there are some action potentials going on when the eye movements, even though it's the opposite side to the stimulus, to the receptive field. And down there, figure D is actually a little different. So in this case, the monkeys has to move their hand, not their eyes, but their hands, and to release a lever. They were keeping their hand on a lever, you can see there, and they have to release the lever when the stimulus, in this case, it's not like the stimulus appeared or disappeared, but the bright of the stimulus changes. So when it changes a little bit, the monkey moves their hand, and then they can perform the task. And you can again see here the cell activity, the action potentials, those lines, and you can see that the eye is not moving. The eye is always at the center of the screen. Then we change to this other experiment, is also with monkeys. And here what's happening is the monkey also has to fixate to look at that white spot, white point on the screen. And again they found out that in this case not only they find the receptive field, but also they found that one of these two stimuli, either the red or the green, they are preferred for the neuron. So the neuron responds strongly to this one of these stimuli. Now in this case it's the red bar. And then the monkey, always looking at the fixation point, they have to make a task. The task consists in saying paying attention at one of those stimuli and detecting if there is any change from one frame to the other frame. So frame 1, the red stimulus is horizontal, and in frame 2 it's vertical. The monkey has to release a lever yes in here, when there is any change. If they remain the same, the monkey does nothing, but if it changes, the monkey has to release a lever. And you can see the action potentials on the right side of the screen. There's a lot of action potential because we are inside the receptive field of that neuron, and also we are choosing the preferred stimulus for that neuron. Then it's normal to find these action potentials. And on the other hand, when you, on figure B, when you ask the monkey to pay attention to what happens to the other stimulus, the non-preferred one, you can see that the action potentials are not as many as in the first one, in A, case of A. And these two images are quite easy. They are just anatomical images. The one is the figure 21.13 is the human brain. They're showing the thalamus, which is inside. You cannot see it from outside, but in this case it's like if the cortex is transparent and you can see through it. And they're trying to show the pulvinar nucleus. So I suggest you to look at this also on some anatomy manuals. And the figure 21.14 is showing a monkey, a rhesus monkey's brain, and they show one specific and very important area related to attention and eye movement, which is the frontal eye field. And they show this because in the next experiment they're going to stimulate, not only register activity, but also stimulate this frontal eye field. In this case, the monkey has to detect where one of the many stimuli on the screen. The experimenter tells the monkey which one. They have to pay attention. If the dimming, the strength of the brightness changes or remains the same. So they set a threshold to see which is the small amount of brightness needed for the monkey to detect. And they found that when they stimulate this frontal eye field electrically with very small stimuli, because if they stimulate too strongly, they will force the monkey to move the eye, make a saccade. But if they just stimulate a little bit, and they can make the detection much better than if they do not stimulate the frontal eye field electrically. And then we go to this final figure from this second video, and again they are showing what we said before. They have stimulation to the frontal eye field and recording from, in this case, before. And what they see in the figure on 21.16 is that this figure 21.16 is showing the what they call, it's an histogram based on action potential. So you have many action potentials on many trials, and the average, they make a very small group of 10 milliseconds action potential and they average them, and then as a result you can see this histogram. You can see difference between when they do not stimulate the frontal eye field, which is the black color, as compared to when they stimulate the frontal eye field, which is the red color. Remember that they are stimulating one brain region, which is the frontal eye field, and this figure we see here is from the V4. It's a different brain area, but it's directly, you can see here, correlate to stimulation in the frontal eye field. And that's it for this second video. We'll see you in the third one.